right, it's time 1 minute past 2, so let's go ahead and get started. We have the whole workshop about about Mariner package for HiC data analysis. Eric's got the rest will be presented, so we go ahead. Thank you.
Hello, everybody. Make sure we've got everybody on.
You can go.
Can everybody see? Hi, everyone. I'm Eric. I'm a postdoc in Doug Phanstiel's lab at the University of North Carolina at Chapel Hill. And today I'm going to talk about Mariner, which is a package to explore HiC data. I'm going to talk about how you can use Mariner with DESeq2 to perform a differential looping analysis. And then finally, I'm going to talk about the plotgardener package, which was also made by our group, which allows you to visualize your genomic data in a multi-panel figure. So if you're not interested in these things, then you're in the wrong room. So before I get started about the workshop, I just wanted to take a moment and acknowledge all the people that are behind the software. So this is the Phanstiel lab. This is our advisor, Doug. Nicole here is the lead developer and leader of the plotgardener project. So a lot of hard work making that happen. This is Marielle. Her data is going to be shown as our test data for this workshop. Sarah here, who's been contributing on all these things as well. And Manjari, who is a visiting professor during a lot of this development, has given great feedback on the project as well as the whole lab. Great group of people to work with. Couldn't have asked for a better team to work on these things with. Also, huge shout out to the Bioconductor community for answering any questions that I had along the way and being very responsive to all these things. So we're going to go ahead and try and launch the workshop at this point. So if you're navigating here, it's the very last one listed here next to the other space themed things. Go ahead and hit launch, and you can navigate to active workshops here. Oh, I have a previous one. I'll stop that. Stop that one as well. And we'll wait for that to show up. But while we're waiting, I'll just go ahead and if you want, you can navigate to this address here and follow along with the pre-rendered material. So I'll go ahead and start there while the workshops are loading. So in this workshop, I'll skip over some of the previous material here and background. Just a little overview. First, I'm going to talk a little bit about what HiC data is. How to interpret that data. I'm going to talk briefly about the workshop datasets that we're using. And then this is sort of structured into three major parts. One, using differential looping analysis with DESeq2, aggregating interactions with Mariner, and then visualizing with plotgardener, which I know is a big draw for people who are not necessarily here for the HiC. So through the course of this workshop, we're going to understand how to work with interaction data in R, learn which Mariner functions to use for extracting different types of HiC data, how to use the successor functions that come with these packages in order to get information out of these objects, and then how you can integrate these things in order to programmatically visualize your data. So, and as I mentioned, we're going to do differential analysis with Mariner and DESeq, make your aggregate plots, and then visualize these. So I have a little Quarto presentation here, just talking about how to interpret HiC data. So you can follow along here, or I'm just going to make it a little bit bigger so it's easier for us all to see. Biologically, HiC data tells us about how any two regions in the genome interact genome-wide. So if we pick any two locations, say these two anchors, the blue and the yellow anchor here, we can trace. These are what we call anchors in the HiC world. We can trace up where that path interacts with the other one, And then this point where these two intersect is what we call an interaction. When we add HiC data, that tells us about interactions for any pair of loci. So for example, this point here tells us about, if I can draw straight lines, the interactions of these two anchors here. And what you might notice about the interaction that I've highlighted in the center is that it's got a high amount of contact frequency, which is what's shown in these boxes. Dark blue meaning high contact, and the lighter color meaning low contact frequency. And there's a lot of high contact frequency in this box compared to its local background, and this is what we would call a chromatin loop between these two anchors. Another way that we commonly visualize HiC plots is in this square matrix heat map format. So we can take our baseline axis there, and then we can rotate it up vertically here so that the start is up here and the end is down here. So as we move down the axis, we're increasing in genomic coordinates, and this way we're increasing in genomic coordinates. And on the vertical axis is what we like to is where we typically put our anchor 1, and on the horizontal axis is where we put anchor 2. If we pick a point along this axis somewhere near the start for anchor 1, and then pick the same exact point along anchor 2, and find where these 2 points intersect, this represents the interaction between those two genomic loci on a HiC map. We can do this for another point, so it's maybe centered at this first blue anchor, and we can see where those interact, and that represents the interaction between the same position. And we can do that for points towards towards the end as well. And we can imagine doing this for every point of self-interaction along both axes, and this is what we would we could draw a line through these in what we would call the diagonals. This is the HiC diagonal. We can fill out this box because these axes are the same whether they're drawn on the left or the right or the top or the bottom. And now we can look at how two different anchors are interacting instead of just the same points on both axes. So let's say we start with anchor 1 and we draw a line from the blue anchor here, and we draw another line from the yellow anchor. This point here represents the interaction between those two anchors. One thing you'll notice is that in linear genomic space, the blue anchor is, we've used the blue anchor before we've used the yellow anchor. The blue anchor is closer to the start than the yellow anchor, and this puts the point in what we call the upper triangular of the matrix. Now, if we do the opposite, if we start with the yellow anchor and then do the blue anchor, this puts the point in what we call the lower triangular. So this is how you can tell what part of the HiC matrix you're on. It's all about which anchor is listed first in the interaction. This is an important concept to note when you're pulling complex sets of interactions. Any questions about that? Another thing about HiC is that it's kind of like an image. Both of these images here contain the same amount of data, the same total amount of data. But what's different about them is the one on the left is lower resolution, while the one on the right is higher resolution. And this comes with how the data is separated into bins. The one on the left has a large bin size, so all of the data, still all the same data, it's just the bins are larger. We can't tell the difference between closer points in space here. Where on the right, we have a smaller bin size, so we can see a lot more detail. And so this analogy holds for HiC data as well. So this map here has pretty large bins. Each bin here is 25 kilobases. So in this square highlighted region here, we've got a 75 by 75 KB slice of data. One thing I should also mention is that I noticed while looking at this map, the diagonal as I've drawn it before was down this way. We can also flip the axes like this so that the start now is here and the end now is here. So same concept, we've just flipped the axes so that the origin here is now in the bottom corner. So just a quick change in visualization. But in any case, we now have 25 KB pixels in this 75 by 75 KB region. If we decrease the bin size here to 10 KB pixels, now it's the same amount of data, but we're slicing it into smaller bits, and we can tell a little bit more about where this loop is located. And we can do this again for 5 KB pixels to see the greatest amount of data. So why wouldn't we go as small as possible, right? That's maybe a natural question here. And part of the answer is sequencing depth and cost, right? It costs a lot of money in order to generate sequencing maps that are deep enough in order to see at lower resolutions. There are other reasons why we're limited in the size that we can go down, but that's a big one. That's the end of the overview of HiCs section. If anyone has any questions about that, if not, I can go ahead and move on. And there's a little terminology section. So if any of that was confusing, or if you want to go back and reference, this is sort of just my off the cuff definitions of these things, not necessarily official or agreed upon by everyone, but this is sort of how I've used these through the software and through the rest of this. Yes.
What's like the average resolution of data sets these days?
That depends a lot on who's doing the HiC.
Okay.
In our lab, we tend to go a little bit deeper. We're a HiC lab, so we do a lot deeper sequencing than most. So we tend to use about 5 to 10 KB. I think that's a pretty good standard for seeing things like features like loops, for example. And while we're here, I'm just going to check on the workshop and see if it's updated. Huh. Should I have signed in to a different thing here? Mm-hmm. This is a different workshop. Yeah, I think I'm on some generic sign in here. Okay.
Yeah, just could be using someone else's.
Yeah, let's avoid that. Let's see. Ah, yes, that would explain it. How to sign in here? Let's see. I don't know my password. I'd have to Thanks for trying.
see like whether you can. Wait a minute. So where are you starting your message of what like that?
Okay, well, rather than sit here and try to get that to work, I'm just going to use the pre-rendered version. So if anyone wants to, if anyone has the workshop, feel free to follow along, but I think this is just as good.
Do you want my link? Because I have it up and running.
That's fine. Thank you. I appreciate it. I think everything is just the same executed in here. Okay, so I'm just going to briefly talk about the workshop data sets before we get to the actual code. So the data set that we're using in this workshop comes from the very talented Marielle Bond, a graduate student in our lab. What she did was she used deeply sequenced HiC performed deeply sequenced HiC among other assays in K562 cells before and after treatment with PMA for 0, 6, or 72 hours. She also performed RNA-seq, ATAC-seq, cut and run for a lot of other factors, but we're not going to touch that data here in this workshop. A truly massive effort. The HiC data was processed with a modified version of the juicer pipeline, resulting in HiC files for each biological and technical replicate. And then we used a loop caller called SIP in order to identify loops for each time point. So since this data set is very large, we'll be working with just a subset of the data in the workshop. So everything's been filtered just for chromosome 1. And just a few of the replicates are being used here. So in total, there are 6 HiC replicates. We're just going to call them replicates. We're going to ignore any biological or technical designations for this. 6 of them belong to the control set. So this would be 0 hours of treatment. And then the other 6 belong to the 72 hours of treatment with PMA. So the loop files and HiC files have also been filtered for chromosome 1. So we've got loop files for 0 hour, 6 hour, and 72 hour. And if anyone is running the workshop, there's one of the first code chunks. If you can go ahead and hit play on that, it's going to go out and download the data from the S3 bucket where it's hosted. Should only take a few seconds to download there. While that's loading, let me know if anyone's having any trouble getting that data. So a first part we're going to start with is the differential analysis with DE-seq2. So here we're going to use DE-seq2 in Mariner in order to identify differential loops between these 0 and 72 hour conditions. And so the first step in this is something that we do called merging redundant loops. So because we have loops called in each of these time points, and due to technical and biological variability, loop callers can sometimes identify slightly different pixels as loops in different data sets. And sometimes this might represent a true biological shift in the loop anchors, but more often this is due to noise and that technical variability. So it's important that we are able to identify these clusters of loops and select one of them to be representative, because testing each of these interactions is going to reduce our power to identify differences in looping. So this figure here is just showing that there's sort of 3 loops called here, and these 2 have quite a few loops called in each cluster. And so what we want to do is select that so that it's just 3 loops in each set here. So to combat this, we can use Mariner functions in order to do this. So the first set of code here is reading in each of these loops. So we've got 0, 6, and 72 hours, and then we're going to assemble them into a list-like structure, or not list-like, a list structure. And then we have a helper function here called as GInteractions, which we can apply to this list. And this is going to convert a loop list to a GInteractions object. So is everyone here familiar with, or rather, how many of you are familiar with GRanges? Most all of you. And how many people are familiar with GInteractions or interaction set? Less. Okay. So a GInteractions object here's the summary of that loop list here. So for each of these time points, we have a GInteractions object, and here we're just selecting the first of that list. So a GInteractions object is essentially a GRanges object that's paired. So here we have one anchor, just like we showed in the previous image, and then here we have the second anchor. So that's how we represent interactions in Bioconductor. And this package was developed by Aaron Lunn. In addition to having the pairs of interactions here, there's this vertical bar where we have additional metadata. So these are what's called the mcalls slot, if you're familiar with that in the Bioconductor types of classes. So here we can store any kind of data that we want that's referencing information about each of these interactions. So here we have things like this AP score average, which in this case we're going to use as a measure for our loopiness, or how loop-like an interaction is. So in order to carry out this clustering and merging, we have a single function for that called merge pairs. And the way that it works is, let's say you have these sort of clusters of loops that are identified in your data set, where the saturation of gray sort of represents the maybe some column or in your data set, like that AP score average that I mentioned. If we provide a column, so pixel intensity, AP score average, something like that, what it'll do is when it clusters these, it'll select the column that's maximum or minimum, depending on what you choose, for that feature. So it's picking the gold highlighted interaction in each of these. If you don't provide anything, it will do the best it can. It will look for loops that are repeated and select preferentially the ones that are occurring more often. In a situation where there are ties, it will take the average of those positions to find an anchor in between. And for downstream analysis, you can then assign those to a specific bin or snap them to a bin as you need to. But more on that later. So this is the code chunk that shows us how to use our merge interactions. We pass in our loop list, define a radius, how far around to look. And this is using the dbScan algorithm in order to do this clustering. And then we pass in a column here, in this case, AP score average. And so now what we've done is we have this merged loops object, which is an extension of the GInteractions class called merged GInteractions that contains your interactions. This function works exactly like a normal GInteractions object. All of the methods are accessible to it, but it also contains additional information about how your loops were clustered. So I'm going to scroll down here and go to the first sort of exercise here, which is finding de novo loops. Yes.
I have a question. Is there an easy way on this kind of object, I've never worked with interactions before, to actually select a specific position that you want to look at?
Specific position, you mean a specific interaction?
No, like a specific position that all the interactions that have the position.
So select one anchor.
Yeah.
So if you're familiar with GRanges, you can do a subset by overlaps. That's a method that's defined for these objects as well. So you can, pick an anchor region that you're interested in, and subset by that to get all interactions that fall within that region. Okay, so because each of these loops were called in different data sets, we can use that in order to find which loops are newly appearing. So from this object, we can use the sources successor to see our 0, 6, and 72-hour set of loops, or that they were used in here. If we want to access a cluster for a particular interaction, so we could say, here's the 1500th interaction. We can use the cluster successor, and this will return a list of data, data tables that has each interaction that was found in that cluster. And the last column here is referring to where that cluster was found. So this particular loop had 3 interactions that were present at 0, 6, and 72 hour. If you have metadata that you want to aggregate, you can use the ag metadata function. So let's say we want to get the mean of the AP score average for each of our select loops. We can then do that and add that, and that will be added here towards the end. And then we can also use the sets function. So if we have 0, 6, and 72 hours, we can use the sets to tell us how many loops were found at 0, 6, 72 hours, and every combination of those 2 time points. So 0, 6, 0, 72, and then all 3. And we can visualize these in an upset plot. So like this. So we can run these code chunks to visualize how many loops were found in each set. The sets function also comes with optional parameters, include and exclude. So this allows you to get just about every combination of loop sets that you might want to. So here we're including 72 hours, but excluding 0 and 6, meaning the 692 is the same as the 692 of this set. So just 72. So 72 excluding the other sets. But we can leave out some of these parameters. So for example, we can just say exclude 0 hours. And what this means, it'll return any loops that they may have 72 or 6, but they definitely don't have 0 hours. So this allows us complete flexibility in our ability to retrieve these different sets. And so as an exercise, how could we use this code to find transient loops, or loops that appeared after 6 hours and dissolved after 72 hours of treatment?
You just exclude the 6 hours and include only 72 hours on the middle.
Yeah. So including the 6 hours, excluding the.
Yeah.
So those are transient ones, right? Exactly. Okay, so that's. I just wanted to mention that as a type of thing that you can do with this. But now to get to more interesting things, which is finding differential loops. Using loop callers to identify when loops are appearing is not always the best method, right? We're relying on the threshold of the loop caller, and it's not a very statistically rigorous method for assessing changes in looping. So another way that we can do this is using DESeq2 in order to find which loops are changing in different sets. So it's typically used for RNA-seq analysis, as many of you know, but it actually works pretty well for finding differential HiC interactions. And it requires 3 pieces of information to run. One is a count matrix, where each row represents a loop, and each column represents a HiC file. 2 is a data frame called call data that contains sample-specific information. So that's regarding your HiC files. And then 3 is a design formula that describes the model that's to be tested. And we can use Mariner to create this count matrix by extracting contact frequency between interactions for each HiC sample. And to do that, first we're gonna start with creating that call data here. So one of the data sets that came with this that you downloaded earlier is a sample sheet. So this sample sheet has information about each of the 12 HiC files that we talked about earlier. It's labeled them sample 1 through 12. The first 6 are control, last 6 are treatment. And then we're just labeling them 1 through 6 for each replicate. Other information about the PMA treatment and the file path is actually what we're going to use here. So this describes the path, data, HiC, and then each HiC file. So that's what this samples data frame looks like here. Since we want a count matrix, we want a single value for each interaction in HiC pair. And we're going to choose 5 KB as our bin size or our resolution for HiC data. And what we'll use is the pull HiC pixels function in order to carry this out. So here we're defining a variable called HiC files, which is just taking that file path and converting it to a character vector. And then using the pull HiC pixels here, we can pass in X here is our merged loops. So that's our G interactions object that contains all of the interactions that we're interested in looking at. The files here contains the HiC files. So just for reference, right, this is on the order of 20,000 loops, 12 HiC files, specifying a bin size of 5 KB, and then a block size. So accessing this data is non-trivial. So we can actually define how much data we want to pull at once. So if we have a large HiC file that's many gigabytes, we can shrink this block size in order to pull less data at a time at the expense of allowing the function to run a bit longer. But if we have smaller HiC files, we can increase this block size so that we can very quickly extract data. So this is sort of a trade-off that it'll, you as a user can modify to in order to fine-tune how you're extracting HiC data. And so if you go ahead and, if people are following along, if you go ahead and hit run, you'll watch it'll go through each block in each HiC file and extract data here. And this is all very sensitive to what type of computer that you're using. I think I've run this on the workshop. It took about 30 seconds to run or so. And then the class that you get here, we're assigning it to this variable pixels. And the object that you get out here is called an interaction matrix. So this is an extension of the interaction set class, which if you're familiar with the G interactions, it comes from that package. Interaction set is a class that extends summarized experiment. So if you're familiar with summarized experiment, then this show method should look very familiar to you. This is extended in order to allow it to contain count matrices in a very specific way. So here we have a count matrix with our, what is that? 2,000, roughly 2,000 interactions across our 12 HiC files. Our metadata here contains information about our bin size, normalization. So here we're using unnormalized data, and matrix, whether we want to use the observed counts or maybe the observed over expected counts, depending on what you choose. And these are all parameters that you can pass to the pull HiC function as well. And to access these things, you can use the metadata or successor function. And then we have one assay here, which is counts. So this is our count matrix with interactions by HiC files. And so to access this, we can just use the count successor. And this is the result of running that. For every interaction in HiC file, we have a single value that represents the interaction between those two loci. And visually, this is just another way of representing this interaction matrix class, where here we have a single pixel. Internally, this data is stored in an HDF5 file, but I'll save the explanation for that for a little bit later when we start pulling larger matrices of HiC data. But to finish running our DE-seq analysis, we can use the DE-seq dataset from matrix constructor, passing in our count data. So using the successor of our pixels to pass that in. Our call data by binding the call data from our pixels with our samples. The samples providing the information about replicate and condition, which we then use in our final design here. So we're doing a differential analysis where we're taking replicate plus condition, and we want to see how loops are changing between these two conditions. And then we can simply run DE-seq with this function, and we get a summarized experiment-like container out, a DE-seq data set. The next code chunk then shrinks the results. Another thing to mention here is the size factors. So it's using the loop counts as size factors, assuming that not very many loops are going to be changing, which in large in part is true. Many of the loops are going to be static between these two conditions. But if you're, yes?
Sorry, I have a question. Like, why do you use DE-seq tool, and did you try other differential expression softwares and how it performs?
I think the choice in software matters very little. So as long as you have a count table, you can use any of the other available packages for that as well. We like DESeq. We know Mike Love.
Yes.
We like DESeq. Very well built software. Yeah, so the last part of my what I was saying was that in cases where you're seeing like total loss of loops, you might want to not use size factor estimations. That's another important thing to mention. So here we can use the summary on our results object from shrinking the log fold changes. And this is just using the default stuff. We can see that there's 24 loops that are up, 11 that are down. Again, this is a very light version of the data set we see. And I think in the final version of this analysis, there were thousands of loops that were changing between these conditions. But with this, just on chromosome 1 in this sort of light sample here, we're seeing 24 up and 11 down. We can then, like any other DESeq, we can make some plots, maybe do a PCA and see that things are separating here in our exploratory analysis. And a really cool thing is we can take these results and we can, because our pixels object is a summarized experiment, we can use the row data accessor to slide the results right into our pixels object. So now, not only do we have our count matrix, our interactions. If you were to run the interactions successor on pixels, you'll get those original interactions out. But we also have all of the DESeq information for each of those, which in the next section we can use to do filtering for gained and lost loops. Before I move on to that, does anyone have any questions? Great. So the next large sections here are about aggregating HiC interactions. And there's sort of three major types of aggregation that you can do. So one is aggregating loops. So if you have interactions that you're interested in piling up, for example, you can do that. Maybe you have domains, so larger topologically associating domains that you want to sort of superimpose and pile up there. Or maybe you have boundary elements that you want to see how they're changing around those locations. So there's three different ways to do it. And the way we wrote Mariner was to be really flexible. Rather than giving you a single function for doing each of these things, which we also do, we also wanted to show you how you could do this with the functions that we provide yourself. So if there's any, oh, this function does almost what I want, but not quite, we want to make sure that that doesn't happen. So you can sort of make your analyses as customizable as you want. So here we're going to talk about aggregating or pulling count matrices here. So before we were pulling a single pixel for each interaction HiC file, but what if you've got an interaction that's pretty wide and you want to see an entire count matrix for that interaction in that HiC file? So for that, we have a function called pull HiC matrices. And the way that it works is for each interaction, you have the first anchor, which I showed you earlier, and then the second anchor here. Each anchor has a width which spans that G range, right? When you provide that bin size, that sort of tells the interaction how to subdivide it into HiC pixels. So for example, if the width was 5 on anchor 1 and the width was 5 on anchor 2 and the bin size was 5, you're getting 1 pixel. If the width is 10, as shown in this picture, for both anchors, now with a bin size of 5, you're getting 4 pixels. So you get 2 pixels per anchor in 2 dimensions for a total of 4. So you would get out a 2x2 matrix for this. To make this easier, rather than having to adjust the widths of everything, we have a helper function called pixels to matrices. So if you already have loop pixels that you're interested in, and you want to see maybe 5 pixels on either side of those loop pixels, you can pass in. So here we're using the interaction successor from the pixels object in order to pass to this function with a buffer of 5. And what that's doing is it's taking that central pixel and it's expanding around it. It's expanding your ranges, your interactions, so that you get, here we're saving it to a regions object. This regions object is now appropriately resized so that you're pulling an 11 by 11 matrix here, 5 on either side of the central pixel for a total of an 11 by 11 matrix. And then you can use the pull HiC matrices function in order to extract those matrices or those regions that you just defined from each of your 12 HiC files at a bin size of 5. And again, you could change this bin size in order to change the size of your matrices, because the regions that you're defining are being subdivided by the bin size that you're requesting, just like we talked about with the image resolution earlier. And again, block size for controlling how efficient this is. And here we're saving it to a variable called matrices. And the class that we get out is called an interaction array. So the previous one was an interaction matrix because it's just a 2 interaction by HiC file matrix. But this is a lot more complicated. It's an array. It's actually a 4-dimensional array. So stay with me here. This is an array. This class also inherits from summarized experiment. But now we have a our 2,000 or so loops, 12 HiC files, and we have 11 by 11 count matrices for each of those. The rest of this object is exactly the same. We can use the same count successor on this object, and now we can take a closer look about what we're getting out here. This array, here we have the dimensions listed here, 11 by 11. So those are the first 2 dimensions. Then the number of loops, the 3rd dimension is loops. And then the 4th dimension here is HiC files. And this is depicted in this image here, where each row is an interaction like before. Each column is a HiC file like before. But instead of having a single value here, we have a whole count matrix. The way that you can look at these things is by looking at the way that if you're like me and sort of like overwhelmed by a 4-dimensional array, then an easy way to look at this is to look at the way it's represented in R. So here the very first this first index position represents the rows, 2nd index position represents the columns, 3rd index position represents this whole matrix here. So that would be the first loop or the first interaction. And then the 4th position here represents the HiC file. So this is the first interaction of the first HiC file, the 2000 whatever interaction of the last HiC file here. Is that clear enough? And here I'd like to talk about.
I have a question about that, actually.
Yeah.
Are there ways you can extract what the different dimensions of the matrices are? Like, can R tell you that, or do we have to know that?
So yeah, if you use on the matrices object, you can use the dim method on that, and it will return the dimensions here. But you can also just look at the show method of this object, and it will tell you that this is how many interactions, files, and things there are. Because this is a matrix, you can just use square bracket indexing in order to grab whatever you'd like. So if you wanted the first 5 interactions, you would do comma, comma. You wanted the whole matrix for the first 5, you would do comma, comma, 1 through 5, comma, 1, or comma to get all of them. Sorry, I wish I could type that for you. And this is one thing, important thing to note here is that this is a delayed array. So all of this data was pulled from the HiC file and processed and put into an HDF5 file. So none of this is actually in memory in your R session, but you can work with it exactly as if it were in memory. So you can do work on much larger HiC files than you can hold in your actual computer's memory. But it's super easy to work with. So if you haven't checked out the delayed array package, you should definitely do that. It's awesome. And then we can, once we've extracted this data in our matrices object, we can use the ag-HiC matrices function. And so we can take our interaction array here, and we can tell it how we want to aggregate things. So for example, if we say by interactions here, we'd get a delayed array with 2 count matrices out, 1 for each interaction, where it's aggregating across each of these HiC files. If we did by files, for example, we would get 3 delayed array matrices out with, or a dimension of 3 with a HiC file, with the HiC files aggregated. Sorry, 1 for each HiC file with the interactions aggregated here, shown vertically. And then if we leave it blank, it's going to aggregate all 6 of these together and give you a total aggregate of all of your interactions. So here, what we're showing, if we do this on our actual data, we can see ag-HiC matrices. We pass in our matrices, and we say by files. And now we have our count matrices, 11 by 11, and we have 12. So all of the interactions have been aggregated for each HiC file. And if you wanted, you could do it by interaction if you want to. And because these delayed arrays work like regular R arrays, you can do things like apply functions to them. So here, I mentioned earlier that the first 6 HiC files are control and the last 6 are treatment. So what we can do is we can index. So here's an example of indexing. We're taking, of our aggregated object, we're taking 11 by 11 and then 1 through 6 here, holding these first 2 dimensions and then summing them so that what we get out is a single matrix for all of those interactions. And then this is control, and then doing the same thing for treatment here. And then we can use the plot matrix function in order to visualize these. And so this is what our HiC data looks like at this point. What you'll notice is that there's a artifact here in the bottom left corner. And this is because sometimes interactions are, oh yes, sorry, question.
So when you're plotting those 2 panels separately, are you able to specify the range of the color scale to make sure you're looking exactly the same thing across?
Yes, absolutely. So good point. As I plotted them here, they're just, plotting to the max and min. So there's no consistency being applied here. In just a few more minutes, I'll show you how to do that. Okay. Good question. Yeah, so to deal with this artifact, when loops are really close to the diagonal and you're pulling, 5 pixels around it, if the distance between those is less than 5 pixels, you're going to grab that HiC diagonal that we talked about earlier. And those are really high counts. Sometimes you don't want to include those in your aggregate plots. So one way to do that is to remove short pairs from your analysis. So that's sort of a limitation of using this type of an aggregate, this type of pileup analysis. This is called an aggregate peak analysis, APA, if you're familiar. And so here is code that we can use. This is actually recapitulating a lot of the steps we just did. We're taking our regions and we're removing the short pairs from them then. This is an endomorphic transformation where we give interactions in and we get interactions out, but now the short pairs have been removed. And then we can do pull HiC matrices here on our HiC files. Same, everything else is the same. We're reassigning matrices here. We're aggregating again and then plotting these. And when we do all of that, we can see that we've now eliminated that diagonal artifact. But this is still not showing us what we want. We actually want to separate our control and treatment by gained and lost. So we want to see the differences between these 2 sets to confirm that our differential analysis was correct. And to do that, we can then separate these. So this is a overwhelmingly large code chunk, but I'll walk you through what's happening. And a lot of these things are the same as what we've seen before. So here we're accessing that row data. Remember, we saved our differential results in the row data of this object. So these are our results. And we're using just indexing in order to select for ones that are significantly up. So a log fold change greater than 0. And significantly down. So these we're going to call gained and lost. Gained in 72 hours or lost in 72 hours. So these reference to that. Then for each one of these conditions, we can look at the gained in the control HiC, and we can look at the gained in the treatment HiC, and the same for the lost. So we're now generating four different situations here. And we're using that same apply function that we used earlier, just using a different set of loop lists. Here we're normalizing to the number of interactions in each set, because you might imagine that there's 24 gained interactions and 11 lost, so we don't want the difference in these to impact our visualization. And then we can find a common scale. So we want to visualize the gained loops on the same scale and the lost loops on the same scale. And as you mentioned earlier, this is how we can ensure that our scale is the same. We can set the Z range here to our gained scale. And for both gained control and gained loss. So now these are shown on the same scale. And they're separated by where the left one here is control. So the gained in control, we're not seeing any loops, which is what we'd expect. And the gained in treatment here, we're seeing loops. Same thing for the lost. Here we're seeing loops in the lost set, which we'd expect, and not seeing them in the gained set as much. Again, these very small loops, very noisy data. It's not ideal and not perfect, but hopefully you get the idea of what we're doing here. And so if all of that was too much and you just want to run all of that in one line of code, maybe you don't want the flexibility to try to do other things, or you just want to run something very quickly, you can just use this function called pile up pixels, and it does all of those steps for you. So here we're separating into gained and lost, and then running those 4 different scenarios again, switching out gained and lost with the gained and lost HiC files. Here min pair distance is telling us to filter out those short pairs. So we're accepting anything that's greater than 50 KB. And then if we plot that, we get the same exact plots, which is nice to see. Okay. Does anybody have any questions about that? So I want to touch on one more point about aggregating things, and then I'll move on to the plotgardener section. So I want to make sure we get to that. Yes.
One more dumb question about ggplot objects, so you can then customize and do whatever you want with them.
So these are not ggplot objects. These are, and that's actually another great question. You can plot these as ggplots. These are just matrices, so you can pass, transform those into long format and pass them to ggplot and visualize it that way. Completely flexible, up to you. The plot matrix function is actually compatible with plotgardener. So when I get to the plotgardener section, you'll understand why that is. Okay, so this next section is covering an aggregate domain analysis. So let's say that you have domains of different sizes. So these can essentially be represented by G ranges. And you want to be able to pull HiC contacts for each of these different sized ranges. So this is something that has been traditionally hard to do, to be able to pull data that's not a standard matrix size. And so here, this can be represented by a ragged or jagged array. And so we've implemented that sort of data storage so that these jagged arrays are stored on disk. And just to cover that, I'll step you through this code here. Here we're taking just writing a little impromptu function. That's essentially going to take these loops and convert them to tads. So each loop is represented by 2 anchors, and so we're going to take the start and end of those anchors. Maybe I can annotate here. So we're taking the start and end of the anchors here in order to define this region here. So this is the start of the region and the end of the region here. So that's sort of what we're working with here. In converting those to the format that they need to be. So here we have TADs. This actually accepts interactions or it accepts ranges. So either one is going to be fine. We can filter them for, here we're just doing gained to show you. And then pull them with pull HiC matrices, same thing, 5 KB. And what we get out of this now, because each TAD is a different size, we actually get an interaction jagged array out. So this is our 24 gained interactions across our 6 HiC files, the 6 gained HiC files. And here we don't know the dimensions of the count matrices. These are variable count matrices. Everything else is, it's a summarized experiment like class. It's not a summarized experiment because that is based on a rectangular data container, and this is no longer rectangular. We no longer have set dimensions for each of these. But we can see our HDF5 file here where the data is stored on disk. We can use the count successor to see this data. So again, we don't have dimensions for the count matrices, so it's n by m, 24 interactions, 6 HiC files, and we get this jagged array here. So this would be the first interaction. This array is 55 by 55 matrix, first HiC file. Last one, this is a 45 by 45 matrices, matrix. You can use the dims function to get all of these dimensions. So you can see what the dimensions of each of the matrix that you're looking at. But for a TAD for an aggregated analysis, you imagine we can't just superimpose things of different sizes. So we need to make them all the same size. And we do that with a function called Regularize. So here we're defining the new dimensions of the matrix we want. So we're defining a 100 by 100 matrix here and doing it in blocks of 10. So we're accessing these things in a block processing approach and scaling it to the spread out the values so that they're not so that they're even across the new dimensions that we're using. And when you aggregate them and then plot them, you get something like this, where we're looking at the aggregate of all of the TADs of the loops that we looked at. And again, there's a pileup domains function that you can run. It does all of these steps for you. So it allows you to be flexible if you wanted to do something unique, but also if you just want to run a common analysis like this, you don't have to know all of these different components. And this is what it looks like for the lost and gained. I'm going to sort of skim over the next section because I want to make sure that we have time for plotgardener. And this is sort of looking at a slightly different question, loop boundaries. So before we were looking at loops, then we were looking at TADs, so this whole region here. And now we're going to look at anchors or boundary regions, so this point. So we're centering our plot here, and we want to see how that changes between conditions. It's very similar, but here we're just using the pileup boundaries function to carry out this analysis for us, creating a scale here, and then plotting it. We can also take these matrices and divide them. So here we're getting a difference between the boundaries, so gained over lost, and setting a range, setting our data range, setting a color palette here, because now the previous color palette doesn't make as much sense because this is differences here. And then visualizing it here, passing in our color palette. And now we can see how the boundaries are changing between these two conditions. Again, the data, not important. This is just to demonstrate the technique. Okay, and so the last section here we're going to talk about is plotgardener. And again, this project was led by Nicole Kramer, who was in the lab at the same time as me. And so this was a fun project that we all worked on. It was quite awesome. But what it allows you to do is programmatically lay out your genomic plots and also non-genomic plots. So here we can initialize the plotgardener library. We can create a page. So everything with plotgardener is coordinate based. You start by creating a page with a specific width and height. So here the default units are inches, sorry. And we're defining this 3 by 3 inch region here. We can pass additional parameters to this plot matrix function. And when you pass positional parameters to this function, it now knows that you're trying to plot it in a plotgardener page. So it will add it to the plot that you're trying to do. So here we're saying we'd like to take this first plot and we'd like to put it 0.5 inches from the top left corner in the X direction. The Y, we want it to be 0.5 inches from the top left corner. We want it to be almost an inch in width and almost an inch in height. And we can do that for each of these different functions. Notice here I'm saving the first gained one as gained APA and the first lost one as lost APA. And when we do that, we get an output that looks like this. So while you could use ggplot to visualize these things, this allows you to have a little bit more control over the exact placement of your plots on the page. And then we can add additional things like annotation functions. So we can add, these are essentially heat maps. So we can use an anno heat map legend function. And here we're passing in our gained APA. That contains information about the Z range that we set or didn't set. And we can specify an orientation for that and a position for that, and do that for both the gained and lost. And when we do that, we get sort of a heat map scale, which is 60 here and 88 here. And there are separate labels for plotting text. So here we're plotting 72-hour loops, 0-hour loops. You can change the justification and the rotation of the text. So here we're justifying the center bottom here. Oh, sorry, my screen turned off. Yeah, and then on these plots with the HiC, we can rotate the labels 90 degrees here. And we get nicely labeled plots out. And when you're done with your visualization, you can use the guide hide function in order to remove all of those grids and borders and things. There's also a function to change the default grids to whatever you want. And then you have your fully formed figure ready to be published. This is very useful for making multi-panel figures. For example, and this is just like the tip of what it can do. You can also specify upfront that you don't want guides, for example, and then do this analysis again. Here, I'm just plotting everything that we saw before, but I'm adding additional things. Here, I'm going to add a HiC plot. So there's a plot a square HiC plot, passing in your data, the locus that you're interested in, the HiC specific parameters that you want to see, and where it's going to be placed on the page. And so we do that for both axes of the HiC plot, and we get something that looks like this. So here is the 0-hour HiC, and here's the 72-hour. Since they're mirrored across that diagonal, we can visualize them both in one plot. We added text for each of these positions. We added labels. We added a coordinate track here. And so there's a lot of things we can do with this. Just one caveat here, because you have so much control, it's also possible to make mistakes and to do things wrong and create misleading plots. So be very careful when you're working with this not to plot things in a way that would be inaccurate. So that sort of wraps up the content that I wanted to cover here, but I'm happy to take any additional questions and about any of these things and talk more about this. Yes.
So all of those S4 classes, that like the merged interactions and those things, you've defined them as part of this package, or are they predefined?
Yeah, so merged interactions, interaction matrix, and interaction array are all defined in this package. In Mariner, sorry.
Yeah, of course.
So those structures are not part of the bioconductor ecosystem. They are part of the packaging system.
Those three classes. So yeah, they are. This is a bioconductor package, so hopefully they extend current bioconductor. So the bioconductor class would be interaction set, would be the most used container for interactions and things like that. So these sort of extend that functionality to allow you to incorporate additional HiC files. Yeah, this is feature focused. So it's focused on not necessarily representing a HiC file as a container, but more the loops and what you want to pull from the HiC data. Or the domains and what you want to pull from the HiC data. So that's sort of the approach that this takes, is grounded in that. I don't know if there's any online questions. Question from JP. Curious about comparing different chromatin domains. Wanted to make a differential HiC plot comparing different regions across conditions. How would you use Mariner to analyze and visualize that using plotgardener? So let's see. All right. So if I understand correctly, I think this is sort of talking about this plot here, how to take, so for example, this is one region under one condition, and we can visualize it in sort of a split HiC plot here with the other condition on this lower axis. All right, well, if there's no more questions, I guess we can call it an early end and catch the short talks.
Is there any questions online?
The only one I see is right here. Yeah.
Just want to check.
All right, let's co-speaker.